So welcome. We're going to slowly bring people in and get people settled. We're going to start. We're going to try and start, keep ourselves somewhat on time. We're nearing the end, but I actually really hope that nearing the end means starting anew. Just like this research is nascent and exciting, and at the beginning, this is our first foray at the RN Center into this kind of a topic on the microbiome, but I don't think it'll be our last. We've already had some people express interest in having a follow-up conference on this next year, and as some of you know, BARD has a growing biology and farm program, and we're thinking and hoping that this can be a recurrent event. But we don't only do this, we also do many other things at the RN Center. I encourage you to take a look at our website. On your seats or around your seats is a postcard for our conference coming up in 2 weeks on October 10th and 11th on racism and anti-Semitism, which is going to be a very exciting conference with really some of the most important but also provocative and influential people on these questions. So I hope you'll be able to join us for that. What we're going to do now, first of all, I'm going to just do a couple of thanks and other things right now to get it out of the way so I don't have to do it at the end. First of all, the name has come up a lot this week, the last 2 days, yeah?
You should mention the VRD.
Oh, yeah, for those of you who are interested, if you're a member of the center, we have something called a virtual reading group that meets every 2 weeks usually, where me and my colleague at the center, Samantha Hill, lead a virtual reading group online from people all over the world, reading essays and books of Hannah Arendt, usually about 10 to 15 pages every 2 weeks. And a couple of people who are in the reading group are here today. It's become a great community of people who read these books together, and many of them are not RN, most of them are not RN scholars, although there are a few RN scholars, a lot of students, PhD students around the world working on it who come in. But it's really a wonderful opportunity to find through the internet a group of people interested in thinking provocatively about the world, which is what Hannah Arendt did. And so anyone who's interested in that, feel free to talk to me or my colleagues, or just go to our website and sign up. So first of all, his name has come up a lot. I just want to once again thank Sandy Lewis, who had the original idea for this gathering, who connected me with most of the people who are here as speakers and others, including Martha Carlin and Alan Lewis, who very much, and Monique Schoenhager, who very much helped me get up to speed on this issue to the extent that I have. And I value all of their input in putting this conference together. Tina Stanton, who's the Director of Operations here, is amazing and has made this conference work like every other conference. So I want to thank Tina for everything she did. Along with Tina, Craig Rothstein's here, and he's our media coordinator, and he's made sure everything's worked and really done a great job. So thank you, Craig. And then you've probably noticed a lot of students helping out. We have a great student community here, and I want to thank all of them for everything they've done. This conference could not work. And then Total Webcasting is here. They're going to webcast this. Robert, when is it going to be up? Do you know? So it'll be up on our website tomorrow morning or your website tomorrow? We'll have a link on our website, Craig, by... So if you go to the conference, you go to the Hannah Arendt Center website, hic.bard.edu, and then you go to the conference website or the Symposium website, there'll be a link on the homepage for that website, and you can watch all the talks if you missed one or if you want to go back and watch one again, it'll be up by tomorrow. So we encourage you to use that if you like. So there's going to be a dinner tonight for speakers. You should have information on that. It's at the Beekman Arms, where most of you are staying. We're going to start this afternoon's session now. There's going to be two parts. One is about a 15-minute talk by Dana Stanley, who we very much wanted to bring here, but was involved in some events in Australia where she's teaching and researching and could not make it here, but she has generously agreed to give us a short 10 to 15 minute introduction to her research on the microbiome that she's been doing, very innovative research in Australia. So I'm going to let her speak for a couple minutes. Because it's on Skype and we don't have all the it's going to be hard. We're not going to do a Q&A. We're just going to give her a chance to speak. And then what I'm going to do is bring all the speakers up again in a big line like we did yesterday, but a little different. We're going to have 4 moderators, sort of, and we're going to take turns bringing people out in groups of 2 and 3 and trying to get little mini conversations going. So we'll see how it works. It's an experiment. So Dana, are you there? You hear me?
Yes, I am. Thank you.
Very great. So I'm going to turn it over to you. And here's Dana Stanley. Maybe you can introduce yourself briefly and then give us about a 10 to 15 minute introduction to your work. Thanks very much.
Thank you. Thank you so much for inviting me to talk to you today. As you said, I'm Dana Stanley. I come from Central Queensland University in Australia, and I'm going to try to introduce the topic and current issues in gut microbiota, and just probably summarize what I do in my own research, which is all I can do in 10 to 15 minutes. This is a very big topic. It's a very popular topic at the moment, because until mid-1990s, pretty much about 2000, we were confident we knew all there is to know about bacteria, because we knew, which pathogen is causing which disease, and we thought that's it. It was like microbiology, the whole area was sitting at the plateau and for quite a few decades. But then there was still unexplained difference. So when we look into things that we can see under microscope, and we can see maybe 100 different species, and we try to grow them on plates and grow them in the lab, we could grow only very small percentage of these species, and people just accepted that we do not have media that is nutritious enough and conditions that are going to replicate the natural conditions well enough to grow all of this diversity we have in nature. And then suddenly we had this big breakthrough, which is finding a genetic marker that is very species specific. A number of them were actually found around the same time. Those genetic markers, pieces of bacterial DNA that can act like a fingerprint, something like DNA, parts of human body that are of human genome that are used for forensic DNA investigation. So we can take any sample from nature now, and we can extract DNA and sequence and see immediately about hundreds to thousands of different species, whatever diversity of that sample, even those that we cannot culture. And now suddenly we can see the remaining 90% that we didn't know what they are and what they do. And the area of molecular microbiology just exploded in last about 20 years. You can see number of papers is grown from a number of papers on fecal microbiota or gut microbiota, went from 1 or 2 in about mid-90s to 1,000 in 2010, and now we have about 100,000 papers on fecal bacteria. And we learned a lot of things, and a lot of things were quite a surprise to us. We learned that bacterial cells outnumber our own human cells 10 times. So there's 10 times more bacteria than human cells in our own body, which some researchers suggested that we should be called walking bacterial colony. Proportion of bacteria we can culture in some environments is even less than 1%. In human gut, we know only about 25% of bacteria. Those we can culture and investigate in the lab in lab conditions. The remaining ones were complete mystery to us. We now have new areas, so-called culturomics, because while investigating these bacteria through sequencing, we are now able to actually design nutritional content of media and actually grow these bacteria and then investigate them further. So human has about 20,000 something genes. Microbiota in our gut has 100 times more genes. This means that only 1% of DNA in our body is human. The rest is bacterial DNA. And these genes, each gene makes a product, which is a protein. And all these products that bacteria make end up in our body. They pass the epithelial barrier very easily. So microbiota is biggest contributor of chemicals and metabolites, proteins in our body. And now, after all these years of research using this new technology, we now consider microbiota as another major organ of human body. The roles of intestinal bacteria cannot be touched in 1 hour presentation. So I've just got a little sort of overview here, and they're able to digest food that we cannot digest. They can produce vitamins and hormones. One of the papers published in 2013 was actually first paper that showed that bacteria can make human hormones and human genes, human products of human genes. So basically, they took fecal bacteria from male and gave fecal transplant into females, and females started making same amount of testosterone as males. So that was a first demonstration that they can make testosterone, and then later on we realized they can make other hormones, because they sort of grow inside of human body and they exchange DNA with the host through the process of bacterial transformation. They take piece of human DNA from human dead cells, incorporate into their genome, and next thing they making human testosterone. They can regulate development of gut. They grow epithelial cells, and gut cannot grow or differentiate without bacteria inside. They prevent pathogens overgrowth. There is no immune system without gut bacteria. You have to have bacteria in the gut to train your immune system. About 30 to 40% of our immune system is actually guarding the gut from leaking bacteria. So you really need that part of our life that part of our body to train our immunity. They can play a role in anxiety. They can control stress hormone levels. Those bacteria in our gut that live on carbohydrates can make us crave that piece of cake. They can control number of brain receptors and many other functions that have been discovered in the last few years. One of the most interesting things that have been discovered is that microbiota is a major player in obesity and weight gain. So germ-free animals, those are animals that we grow in sterile rooms, giving them sterile food. So they're completely germ-free. They have no bacteria in their gut or in their body anywhere. So they grow in these bubbles, controlled environment. They need 30% more calories to be able to gain the same amount of weight or to absorb same amount of calories as the other mice. This is because they don't have microbiota to break down the food in the gut. Control mice, so normal mice, will have about almost 50% more fat than germ-free. Germ-free mice that don't have microbiota cannot gain fat. The fecal transplant from obese mouse, and this is that very popular, well-known experiment, the photo from that paper, where they took fecal transplant from obese mouse and injected that fecal material through enema into the very normal lean mouse. And on the same feed, under same conditions, lean mouse became obese within 2 weeks. we can do now the same later on. The research, not willingly, but we played with fecal transplant in trying to treat Clostridium difficile, and we confirmed that if we have obese donor human, yes, we will cure Clostridium difficile in a patient, but if we had obese donor, the patient will become obese. This has opened new view on obesity, because obesity brings a lot of stigma to people. Like, how come you can't lose weight? It is not that simple. We have now identified obesogenic bacteria that can make you obese if you get it. So some people now refer to obesity as epidemics, and it is actually like bacterial type of epidemics. So another thing is so-called microbiota genocide. Another thing that we discovered is that we have completely different microbiota to the healthy human microbiota that is adapted to humans over thousands of years, until our grandfathers, until the big changes in the way we make and eat our food. This is when microbiota started dying off. The bacteria that were really important for our health started to be just like loss of species that we have at the moment on the planet. So Western lifestyle means that today about 50% of diet is refined carbohydrates. We were not supposed to even eat those carbohydrates. Our carbs should be fruit and vegetables and natural products. But the result of having so many carbohydrates is that instead of under normal carbohydrate intake, where we would have, for example, all carbohydrates used up in upper ileum, and then fiber and indigestible material arrives to colon, where bifidobacteria and good probiotic bacteria break it down in these amazing products that regenerate our body. We call them short-chain fatty acids, acetate, butyrate, propionate. These short-chain fatty acids have a role in regeneration, rejuvenation, and everything that's repair of all different organs. Unfortunately, we don't have fiber. Instead of eating fiber, we have all that oversaturation of carbs, and now carbs are coming into the colon where they don't belong. And instead of probiotics, now we're growing pathogens in the colon, those same pathogens that make us obese. So all sorts of issues are coming up from this destruction of this organ through this, as I say it, microbiota genocide. Also food, all sorts of additives and other things that do not belong to food as we are adapted to it. So US stats, we're talking about diseases now that are linked to issues with gut bacteria. Directly linked to gut bacteria. We're talking about by 2030, we're going to see another 76 million of obese adults, almost 8 million cases of diabetes, and so on and so forth. And this situation requires our immediate attention. We need to do something to fix our guts. We eat insulinotropic food. We behave in insulinotropic way, in a way that our chaotic nutritionists have published ridiculous papers suggesting to cut off fat completely so you don't get fat, without thinking that carbs can make you fat, for example. And people were getting even more and more confused. Also, nutritionists suggested, instead of eating like regular 2 or 3 meals a day, they suggested to eat 6 meals a day and sort of smaller meals a day, which meant that your body was non-stop digesting instead of resting, because naturally we should have resting and digesting periods, where digesting periods should be very small rather than 24/7 as what we have now. So the result of all of these things is also major problem in human health that is not that well known, and that is leaky gut. So not just gluten, because we eat modern grain that has 20 times more gluten than normal old grains that we used to have 100 years ago. It is all other food, this type of modern Western food. For example, people that were living 1 month on Western-style diet had 71% increase of plasma endotoxin. Endotoxin comes from gram-negative bacteria. and it should not be in the blood. Endotoxin can be in the blood only if pathogenic bacteria leak from the gut into the tissue. And so you go on Western food for 1 month and you have this leaking of bacteria through the whole body. So we are in constant inflammation from the food we eat. And colonic bioreactors running empty because we do not eat fiber and we do not make regenerative chemicals that are capable of fixing our gut. And we are caught in this circle, which then leads into obesity, type 1 diabetes, type 2 diabetes, like all sorts of issues are linked with these gut problems that I'm talking about. So leaky gut is really major health issues. It comes from feed additives, unbalanced feed, and antibiotics in feed, other medications and chemicals in feed. There is a list of diseases that have been linked, and every single thing in a bracket is a reference, like a scientific paper that directly linked these diseases to leaky gut and issues with leaky gut. So not just that our gut mucosa is leaking, a body chemistry is so out of whack that we have leaky lungs, leaky skin, brain barriers leaking, placenta. When placenta is leaking and bacteria get into the fetus, we have miscarriages. And it's a literally disaster of human nutrition. So, as I said before, it really affects the whole body. So signs of deteriorating health are now referred to as dysbiosis. Basically, that's when we lose our good bacteria and gain pathogens instead. So, but there's no good guys in the gut. Once the gut leaks and lactobacillus gets into the tissues, they are going to have a feast on your meat. They are not going to pass that opportunity. So we need to think about how to reduce gut leakage. Every time you have a fight with your husband or your wife, the stress levels will cause the gut to leak. And imagine with the food that is already weak in the gut, we leak all the time. So probiotics, most of the probiotics that I tested in the lab come from, are actually dead bacteria that cannot grow, because freeze drying process really makes them almost half dead. So you have to grow them 5, 3, 4, multiple passages to actually make them capable of growing before they can actually do anything in the gut. There are products that are better than others, so it's not sort of generalistic, but it is very common that they are actually dead bacteria in a capsule. Gut microbiota of humans is formed by 2 years old. After that, even if you take healthy probiotics, your gut is going to kill any new coming bacteria, or at least attempt to kill them. So we have to think about how our children are getting colonized. We have to try, first of all, we cannot sterilize everything. Hygiene hypothesis is linked to all autoimmune disease. We have to expose them to good bacteria from day one. Of course, major solution is radical change in feed. The other thing that is happening at the moment is fecal transplants. But what people are doing before they go on big round of antibiotics, for example, if you have to take antibiotics, a lot of even researchers in this area will take the fecal, their own poop, freeze it, and when they're finished with antibiotics, they will push it back where it came from and reconstitute their original bacteria so that they do not get into dysbiosis and leaky gut issues. Now, I'm just going to quickly mention, because my time is already well up, the areas of research that I work in. I do a lot of research on autoimmune disease and gut microbiota, prostate cancer, again, in relation with gut microbiota. But I do a lot of veterinary research with Australian livestock industry and farmers that are actually voluntarily removing antibiotics from feed and are facing a lot of challenges trying to actually run a new system economically. And what I'm finding in that type of research, with veterinarian research, is that number of products that people frown upon, such as biochar, bentonite, zeolite, oregano, clove, cinnamon, a lot of these products actually work better than antibiotics when they're used in feed. But there is a lot of sort of distancing of researchers that look into natural products. If it's not synthetic, it's not good enough. Literally, it's like you're selling a snake oil. But then we are trying to change that opinion by showing genomic, genetic, and all other sort of proof that these natural alternatives to antibiotics can really work much better in actually livestock production than what we had originally by using antibiotics in feed. And so I will finish there. I'm glad I was able to kind of cover this topic. Thank you for not interrupting me. I went 3 minutes over, and thank you for invitation. The photo is just photo of my team during dissection day. Thank you very much.
Thank you. I should mention, I think it's 4 a.m. where Dana is, if I'm not mistaken, so thank you for staying up for us. So thanks. I mean, if there's any one quick question that people have for her, I don't know, it's a little hard, but I can repeat it and maybe she could hear it.
Yes, I can hear.
Yeah, you want to ask one? Yes. Do you have a mic, Craig, for her?
Oh, it's just something she said in passing.
Yeah.
I just heard you said something very quickly. Did you say you were working with prostate cancer in some of your research?
Yes. We are running clinical trials on prostate cancer. Unfortunately, in Australia, we still use PSA test, and I'm very, very much against it. America does not allow PSA test. It's almost like 50-50 chance that it's going to be correct. So we have a lot of men that end up that do PSA tests, and it shows high result, and they freak out. And although they have negative biopsy, they kind of decide just to take it out, which works really well for surgeries. But when you look into recovery of those men, it's really difficult. So what we're trying to do is to develop a marker, a microbiota marker, using metabolites that are produced by cancer cells to detect them in fecal material and in urine.
In order to get a more accurate testing, screening, yeah?
Yes. We're trying to find a way for early detection. Unfortunately, we aimed for early detection, but we actually managed to find markers that can predict that patient will get very aggressive cancer, but we still do not have markers that would be early detection as we wanted to find.
Thank you.
We're going to have one more question, and then we'll let you go, Dana.
Thank you.
Are you working with Peter Collington?
Sorry?
Are you working with Peter Collington? Or no?
No.
Okay. We're going to let you go, but I one thing that's really comes across with your presentation and so many, and it's something we've talked about a lot so far, is how little we know about the microbiome and how exciting and nascent this field is and how quickly it's jumped up on us. And you know, your charts, but also what you're working on is very exciting. So thank you for sharing it with us. And we're going to have our last panel now where we're going to bring, if we can, try and move some chairs up and bring all the speakers up here. And we're going to try a little experiment with asking them to have little mini conversations. So keep maybe the 3 middle chairs free if we can, or we can sit in them and we'll do musical chairs. Thank you again, Dana. Hi. So the way I see it is we'll just take turns.
Okay, who's going first?
Either way.
You go first.
You go first.
Well, I have a question
I
I'd
have
like
one.
to ask based on what she just said.
Okay, who do you want to ask?
Well, I want to ask I would like to ask an ag person and a physician
Physicians of all
and
that.
a farmer. All the Daphne left?
Monique? You're going to be on the first question. Sit in the middle. Martha has a question. Rodney? Where's Rodney? Can you come up here?
We're doing Martha, so they can see the other end.
Oh, yeah. Alan and
Inflammation and obesity.
Oh, okay.
Rodney, can you just sit right
I'm just
here?
going to ask, based on what we know, we use antibiotics and statins now.
Art.
We're going to have a physician.
Martha. So the way this is going to work, we have a couple of. Can you bring the mics up actually for the? No, it's all right. We have a couple of moderators: Martha Carlin, myself, Mark Williams Jr., and Alan Lewis. Right, and we're going to create little mini dialogues. So the first one, Martha's going to ask. And she's asked Monique, Rodney, and Art to come up, and she's going to ask them a question.
Because it's interdisciplinary. So we have a physician, we have a toxicologist, research PhD in many fields, and a veterinarian. So you just heard in the talk she gave us about the rise in obesity. And this is more like a thought experiment question, but there's actually been quite a lot of research on something called feed efficiency in animal health, because in order to increase the speed to get an animal to market, you want the feed to be as efficient as possible. And of course, when you are optimizing for feed efficiency, you're optimizing for weight gain. And so one of those tools has been antibiotics in the feed to increase the weight of the animals. So just as a thought experiment, or, A plus B equals C, what's your thought about antibiotics in humans in the gut and how that could be connected to the obesity epidemic, and, from your perspective in each of the different areas, and how that ties to feed efficiency in animals and the inflammation that she talked about?
Well, I'd first sort of like to establish a picture. We all are aware of the obesity epidemic that exists. I particularly specialize in maternal fetal medicine, so pregnant ladies. And the statistics that the American College of Obstetrics and Gynecology are now presenting are really frightening. In some studies, there are about 56% of pregnant ladies coming to their prenatal visit are either overweight or obese. That's a scary statistic. So speaking specifically to Martha's question about whether antibiotics have a role, I am not a scientist, I'm not a mouse, I'm not a horse, but if you look at the amount of antibiotics that we're exposed to based on the information we've received throughout the day, that certainly there may be a role in we're receiving at least 3 courses, I think it is before age 2, 17 before we're 18. I think that's correct, what we discussed yesterday. And we're seeing a significant impact on our microbiome directly, because these are taken in, for the most part, by mouth and affecting directly our gut bacteria in that way, that it may also lead to the obese epidemic that we are seeing.
So two points I'd like to make relative to agriculture. Poultry broilers are being harvested at a younger age. In fact, they grow so fast that they have leg issues. They can't support their own weight, and they often develop ascites, which is fluid in the abdomen area. But just look at the fat pad. If you're as old as I am, you'll remember what a broiler looked like in the old days, and look at the fat pad now. And you can see they're harvesting them very young, so it's not a super obese chicken because it's still so young, but the fat is there and developing. So yes, that's the result of some of the physiology. The other thing about obesity I want to point out is it's important to keep the system in mind. So we have the gut and the nutrition, the microbiota, the one-cell epithelial layer, mucin, and the layer, the immune cell underneath. There's a difference in adipose tissue, and I've had to give whole lectures on it. I won't take long. White versus brown adipose tissue, and the browning or beiging of adipose tissue, where you convert to using more thermal energy instead of storage. So the white adipose tissue, you're just storing it up. And what you may not know is that becomes an immune type organ, adipose tissue does, because cells called macrophages inhabit it. And there are different kinds of macrophages, and white adipose tissue has different ones from beige or brown adipose tissue. You can actually, in experiments with mice and with rats, drive insulin resistance to insulin sensitivity and convert away from obesity and metabolic syndrome by transfusing in already committed macrophages that are associated with brown or beige tissue, the so-called M2. And they change the inflammation factor, and they reverse engineer through the barrier and a microbiota to make this disappear. Now, we can't really necessarily easily do that in humans, but it reveals the fact that if you're working on just one aspect of it, diet, microbiota, leaky gut, barrier or endotoxin across the gut, things that shouldn't be there crossing, and then lots of adipose tissue lining your visceral fat area, keep in mind, the immune system has changed. The immune system has made that an organ itself, and it's made it a macrophage inflammatory organ. And you eventually are going to have to reverse that, or that inflammation will migrate to another tissue, because there is a spread. There is an inflammatory spread out of white adipose tissue, and it's not just dealing with that, it's dealing with what comes next that's affected by inflammatory damage. So,
in addition, just going on that we're also seeing that inflammation affect directly the hormonal axis. So it directly affects, for example, a pregnant woman's ability to achieve pregnancy and to maintain a pregnancy complication-free pregnancy as well.
Well, the first thing I want to say is supposedly, well, we outlawed, our government in the U.S. as of January 1st, 2017, outlawed all low-level feed antibiotic feeding for the use for rate of gain or feed efficiency. And then to try to answer the question, up until that time, we knew low levels of like ASP, aureomycin, sulfamethazine, and penicillin would improve rate of gain, but we never really understood why. Well, wonder of wonders. See, we never really understood exactly why. And the next thing is, when we were increasing rate of gain and feed efficiency, we probably weren't putting on more fat. It was more in, well, muscle mass and protein. So I'm disgusted with myself because I just listened to Dana, and up there when she fed a modern U.S. carbohydrate diet, did she say it took 5 times longer with the carbohydrate to go through, or was it 5 times shorter? Shorter. It was shorter, correct? Yeah, okay, and I should have known that, but anyway, that that's just another thing that, well, gut passage we know in a cow or whatever, if you don't feed them enough fiber, same thing if you're the soil. If we, something we haven't even talked about, we know if we use liquid manure and put it on the ground versus when pigs are on the same diet, if I have some of the pigs on a farm fed the exact same way that are on an indoor-outdoor building with a cement floor where they're using basically corn stalk bales and then hauling it back out to the field. If they haul that manure out and then they have new pitted buildings and haul the liquid manure to another farm that's farther away, we're having better luck keeping our organic matter up where we're putting on the manure with more fiber in it. So, feeding the cows rumen, feeding the soil is very similar.
Great. Actually, I was wondering, Alan, could I have you go sit next to Belinda there? And I have a question because it seems like one of the things that was discussed yesterday that I got confused about was this law that Art just mentioned that said you can't use antibiotics for feed. I guess for big animals. So it was not small animals. Did I get that right? Or is it for all animals?
I didn't want to lead you to believe you can't use them, but you can use them for a medically disease purpose, but you can't use them just for feed efficiency or growth promotion. And so you have to have a veterinary feed directive. We have to fill out a sheet and keep records when we do it.
So I mean, my question is, I mean, I got mixed messages from different speakers. So my question is a very simple one: is as a consumer and as a citizen, should I be satisfied that this law has largely, in an intelligent way, led to a reduction and an adequate reduction in the use of antibiotics in farming? That's my question as a citizen and as a consumer. And it seemed to me that Belinda and Alan and some other people had some different opinions on that. And I just want to hear if we can, help me, because I'm confused about it.
I'm really glad you asked that, and I'm really glad you asked it of Alan and I, because I don't think we're as far apart as people might have come away. And Alan, you can correct me if I'm wrong. I teach judicious use of antimicrobials to veterinary students, and somebody this morning said improper use of antibiotics contributes to antimicrobial resistance. I teach that every use of antibiotics contributes to antimicrobial resistance. And I don't think consumers should be completely comfortable with that all antibiotic use is judicious or necessary, and in veterinary medicine or in human medicine. And while I applaud the change in the law to stop the feeding of antibiotics for growth promotion, there are going to be mistakes made, and some of them are probably going to be deliberate. We've heard today about people doing bad things, and people do bad things. I think for the most part, your typical farm animal veterinarian and your typical farmer does not want to hurt people, does not want to hurt animals, and does not want to hurt the environment. I know one of Sandy Lewis's motivations for this meeting focuses on his own experience with anaplasmosis in his cattle herd that I alluded to yesterday, and to his belief, his own personal belief, that there are people out there that would introduce anaplasmosis into a herd of cows so that they had an excuse to treat them medically. I don't know anybody that would do that. I know large segments of cattle in the South where they get infected with anaplasmosis as baby calves. That organism becomes part of their microbiome and never harms the animals. And 100% of the cows are infected, and it doesn't do them any harm at all. And in Sandy Lewis's farm in New York State, where they'd never been exposed and they got infected as adults, it was killing them. Generally, New York State has been free of that disease, and Texas and Oklahoma, 100% of the animals get it when they're babies because they get bit by ticks that are infected. And it's like it's in an okay state down there. They don't use any antibiotics. The problem is the intersection. And if there's a farmer that introduces it into their herd, they have to worry about all the animals dying. And they reach for a tool that can keep them from losing their family's entire investment. They are not doing it because they want to poison the animals, the soil, the whatever. And so the problem is way more complex. Then there aren't always simple answers. There aren't always simple answers. I would like to promote cattle health to the point where people don't need antibiotics. I would have liked to have been the veterinarian that was consulted when Sandy Lewis was buying those cows, and I could have prevented that problem entirely. And Sandy wouldn't have had that experience, and we might not be having this conference today.
So there were a lot of loose ends that didn't get tied off yesterday during the agricultural panel. And so to bring Belinda's comments probably to a much higher level, let's talk about industrial agriculture and the things that happen in that system where most of our food comes from. First of all, we are applying synthetic fertilizers, which is disrupting the soil health. We're applying herbicides, which are essentially binding up minerals and creating a carrier as opposed to a healthy soil. Those are agricultural antibiotics. You can choose your term, but effectively, that patent that was discussed yesterday is a broad-spectrum antibiotic, which is glyphosate, and that's the active ingredient. There's also a dozen others that have been discovered, including arsenic, which is actually the active ingredient. Then we have these concentrated animal feeding operations. When you put animals shoulder to shoulder, breast to breast, butt to bill, they are not going to be healthy without antibiotics. Furthermore, those require large-scale GMO monoculture. Between here and where I live is 200 million acres of corn and soy, which are GMO, which are subject to multiple passes of Roundup with glyphosate, plus dicamba, plus glufosinate. This is our industrial food system. We have eliminated wetlands, we've eliminated wildlife habitat, and we've eliminated somewhere around 2/3 to 3/4 of our insects. Now, if you're at a microbiome conference related to agriculture and human health, and you're just looking at those 200 million acres, which is the heartland of America, you don't get the feeling that this is a healthy way for us to manage our country. When it comes back to antibiotics, the fact is, and Belinda and I probably agree on this, but I'm going to give you my perspective, because I travel all over the country talking to ranchers and farmers, north, south, east, west, doesn't matter. Like Belinda, lots of conferences, lots of whispering in the back room from all sectors. The idea that the poultry industry or cattle industry is not using antibiotics is preposterous. Fact one, it's profitable. Fact two, we're making and selling massive amounts of agricultural livestock antibiotics. They're going somewhere.
To the two livestock, large animal vets who are sitting on both sides of you, do you have a sense of where those animal antibiotics are going? I mean, you must know people buying them or not.
Go ahead. You go
Well,
first.
He is not all wrong. I don't want to pick on my clients and my friends because they're caught up in this system. But the system is what's broken. He and I agree on that. I mean, but now that we have all these bigger scale systems, partly because of economy of scale, and we figured out how to do quite a few things right, because as you get more and more ahead there, disease pressures are higher, you got more critters in an airspace, and you got to do things better or it will fall apart. So, but now we have all those systems. For us in Iowa or whatever to try to go back, we need more agroecology. I've been saying that all along instead of agrochemistry, but for us to say go back to more farmers, which it takes more farmers if you're going to. We had more farmers before. In Iowa, in our little Iowa communities, what do you have? We have farm policy based on acres rather than people, and so we have a new bank, Casey's, and a gas station, and then almost nothing. And so the smaller people, don't have enough widgets, whether it's gallons of milk they produce, or so many pigs to slaughter, or fat cattle, or whatever we're producing. We're producing enough of them now from bigger outfits. I don't know how you redistribute it, but if you could redistribute it, it would sure be beneficial, because then you could have more crop rotation, and you could have a little livestock on every farm. There's a lot of things we threw away with our capitalistic system with the wrong rules. Our farm policy it's been wrong from the get-go in Iowa. See, it promotes, well, it's always been based on corn base. So if you're a small dairyman or you plant some hay, have a cover crop, oats the year before to get it planted, that all subtracts from your corn base, so you get a lower government payment.
So I'm going to add one question to this, and I'm going to let Ted, and maybe you guys or someone else will answer it, is we've heard a lot about small farms, right, yesterday. If I'm hearing what Alan and Art are just saying correctly, the law may be a well-meaning law, but as long as you're going to have big farms, it doesn't matter. Am I right? And that's what I'm hearing? And does that mean we need to actually break up the farm? So it's just a question. I'm just trying to figure this out. But I'll let Ted go.
Okay, I'd like to answer your question as I go at this. And I guess I'm kind of an optimist. And these new rules, we've had these new rules, they're just coming in. We have a transition period. We have many transition periods in livestock veterinary medicine. We have people like Rodney that have the answer for this, and I'm talking about the people that can build up the immune system. And as we build up the immune systems for the animals that are in our type of management now, you see the use of antibiotics dropping off drastically. We have some hog producers that used to use a lot of antibiotics in the feed, and they were seeing the exact things that Dana was talking about. We were seeing the microbiome, for lack of better words, but this is a take-home message, that they were getting screwed up. And so these animals were still not growing as fast as they thought they should. But when they got the antibiotics out of there, they had a better rate of gain and a more efficient gain. And we heard about some of the farmers yesterday that are doing a number of these types of things. We're on the edge of a divide here, and I sure wouldn't throw in the towel. We have to look at research for immunology, getting that microbiome going, and we're going to have the growth that makes our farmers economical in each species, and this includes swine, beef, and chickens. I'm going to hand it over to Arden.
May I ask a question quickly, since we're talking about this? You know, and this is actually for my own personal benefit as well, talking about human medicine. Does it matter how we give the antibiotic at all? I mean, most traditionally, my antibiotics are given by mouth. I mean, and this is a question, you know, that Sandy has plagued me with repeatedly, that IM or IV seems to be a better route for both humans and animals, or is it just that we're killing our microbiota regardless?
No, wait.
She's a pharmacist, so that it's a pharmacology issue.
It's a pharmacology issue, and that is that the worst cases of C. diff and disturbance of the microbiota happen at the hands of IV antibiotics in the hospital and in the homes. Home health. Horrible, resistant, vanc-resistant C. diff that does, however, by the by, respond to a combination of 4 herbs when used together in the proper doses. One was what she mentioned, olive leaf extract, along with a particular form of Echinacea purpura, artemisia, which is known as Chinese wormwood. And when you put those together, they will take out MRSA, let alone vanc-resistant C. diff. But it does matter depending on the antibiotic. But the IV antibiotics that can be given and are given IV hit that blood flow and absolutely destroy it. And the same with the IM. Furthermore, it's destroying the flow. Everybody thinks the blood is sterile. It's disturbing the microbiota in the bloodstream and the endothelium, the endothelial tissue. We have to have those bacteria for that as well.
So no safe way.
No safe way, honestly.
So I want to answer the question on the egg thing. Do we have to be big or do we have to get all the small ones? I've consulted on some of the largest farms in the world, over the last 20 years, from 110,000 acre operations in Australia, actually stations, which are 250,000 acres in northern Australia, to some very small farms, to greenhouses, and you name it, and everything between. The reason that we have antibiotics really in the system is because of the dietary process in the animal operations today. If you look at Organic Valley, how many of you are familiar with the company Organic Valley? Yes. They are one of the largest organic co-ops in the United States, about a $500 million company. Up until a year ago, Paul Detloff was their consulting veterinarian. He's an organic vet. He was a conventional vet for about 3 decades in Wisconsin. And then enough of his farmers converted to organic that he converted to being an organic vet. And as a result, he visited every single farm that Organic Valley has on their milk truck. And some of these farms are small. Yes, they may only be 20, 30, 40 cow herds in northeastern United States, or they may be 5,000 cow herds in Northern California. They are still organic operations. They are not using antibiotics. But the key is not just the use or non-use of antibiotics. It's that they have changed the diet of the cattle. And the first thing that they do is they reduce or get rid of, well, one, they got to get rid of the GMOs, but they get rid of all of this grain in a rumen. If you know anything about how a rumen operates, you cannot expect the rumen to be healthy and feed it grain.
Can you tell me what a rumen is?
Rumen ruminant animals are dairy cows, beef cows, deer, and so on. So they have multiple stomachs, if you will. And so they have to have a grass. They're supposed to be eating grass. When we put grain into there, we drop the pH of the rumen. We cause an acidotic condition, which then hammers their immune system and makes them susceptible to all kinds of infections. They're highly inflammatory. So we can do it with commercial agriculture, but we have to change, one, the nutrition in the soil, because that's the first thing that Dr. Detloff does with all the dairy farmers is he changes their soil program. And number two, he changes their diet. That is the key thing that's completely not been addressed here. We have the solutions today. We have a man, well, Don Huber, but his very good friend, Michael McNeil in Iowa. He is a consultant. Over 50,000 acres of corn and soybeans on commercial operations. One gentleman has 15,000 acres. He converts a section a year to organic. And on their own operations, their organics outproduce, outyield corn and soybeans per acre than their conventional. So this is same farm, same farmer, same climate, everything. The organic outyields the conventional, and they have better weed control, and they're not using any herbicides.
Great. Alan, are you okay with large-scale organic farming of this style? I mean, I heard you saying you don't like large-scale farming, so I just want to hear.
I'm scale neutral, but let me finish my thought on antibiotics. They're being made. 2 things. The world is flat. Half of our beef comes in from overseas, especially Brazil, New Zealand, Australia, Uruguay, Argentina. Now, it's all labeled product of USA because that's how our government rolls, right? Right now, in Brazil and China, they are using colistin as an antibiotic growth promoter. Colistin, notice the shaking head, is the antibiotic of last resort for MRSA infections. 100,000 people in this country this year will die of MRSA because it's untreatable. The doctors will tell you, I can give you colistin or you can go home and die. It's likely to wipe out your kidneys and you will not survive anyway. We have a flat earth. The second thing, and I'll be brief, but what you hear from Purdue, Pilgrim's Pride, Tyson, especially in the poultry industry, is we do not allow subtherapeutic use of antibiotics. Well, all their birds are sick. All they need is somebody to say, these birds are sick, so we're going to give them antibiotics. it's preposterous to think that we're making all these antibiotics for the ag system and they're not being used. Now, what I hear on the dirt, across the country is, yeah, we don't use antibiotics. The driver who drops off the feed puts a 5-gallon bucket of white powder and says, hey, put this in your feed today. It's not on their books, it's not auditable, it's not searchable. But if you don't see never ever given hormones, never ever given antibiotics from a trustworthy meat provider, I'm going to tell you, you are more than likely buying meat from animals that were treated with antibiotics that are creating antibiotic resistance genes that's flowing through the manure and the urine and back into our global biome. Scale neutral. I know big ranches under American Grass-Fed Association, they have 1,800 ranchers, 100% grass-fed, 0 antibiotics. You use antibiotics, you failed, you take that animal off the herd, they don't get the seal. You can scale this up. I know a herd with 6,000 head of cattle down in Arizona. They're just moving them around every day, making sure it all works and staying healthy.
Belinda, isn't there testing for, like in the poultry industry, for the, from what I've heard from similar producers, is that they have developed, they'll say, they have similar environment for the animals, but they'll say this is going to be antibiotic free. Take away the antibiotics and the sick birds. If a house gets sick or individual animals get sick, then they move them on to another product line. So the regular product line, and then they only keep the healthier birds in the antibiotic-free line. But I guess I'm interested to speak to that because I know that some of the veterinarians I've spoken to who work for these companies, such as Purdue, they've made some positive changes in those houses, more in line with the organic production to grow healthier birds. But there's also it's been very disruptive in the veterinarian industry to see these animals suddenly have to make this significant shift in their diet and health. But I think also for some of these companies that are going antibiotic-free, there is also certification to say that these, like in the dairy industry, that there are not antibiotics in this meat. But I guess I'm wondering, is that effective? Because I'm sure there's
effective for what?
To ensure that meat is genuinely antibiotic-free. I'm sure there are cases where somebody's dropping off a bucket of antibiotics, but I don't know how widespread that is. And it seems like there is some additional testing of the meat to ensure that it is what it claims to be.
Yeah, so whether there's a residue in the product isn't, I don't think, the issue that people are getting at here. So what we're trying to avoid is supporting antimicrobial resistance. And the antimicrobials that are used legally, the animals are treated, and we know how long the antibiotics stay in the body, and we wait until the antibiotics are not there. And yes, the meat is tested, and yes, every truckload of milk is tested, and we can be relatively confident that there's no antibiotic residue. The residue is a very, very, very small percent. And I would say that most of our food in this country has no antibiotic residue. And yes, the antibiotics that are being produced are being used in animals. There is no doubt that when a truckload of animals goes to a feedlot with 100,000 animals on it, that you are commingling disease, just like if all of you were packed into a coliseum with 50,000 other people, in 3 to 7 days, people would start getting sick. So we have systems that are not health friendly. And they have developed because of economics. The farmer who owns that feedlot doesn't like sick animals, and they learned a long time ago that their tool was antibiotics, and that they could make money because the corn is plentiful, the feed is plentiful, the hay is plentiful, whatever they need is plentiful where they are, and they have gotten away with fixing the sick animals. You can use a lot of antibiotics in a short period of time when there's 200,000 animals that weigh 1,200 pounds. It doesn't take long to use way more antibiotics in that small place than it would take for all the people in this room to consume an equivalent amount of antibiotics. So when we talk about 85% of the antibiotics being used, it doesn't take long for one cow to consume a lot of antibiotics, even being treated once.
So where does that meat go from the feedlot? And that's a part of the food chain we haven't talked about here, is where that meat that is, so we've talked about how we can avoid it and organic and all of that, but there is a large portion of the supply chain that still is from cows that have had antibiotics. Right. And where does that food go is a conversation we haven't talked about.
Right. So,
and it goes into some of the cheaper food supply.
So if that animal gets the antibiotic, and then they hold that animal for the prescribed period of time that's on the label of the antibiotic, which may be 4 days, it may be 30 days, it may be 60 days, depending on the antibiotic and what it is. After that period of time, it is illegal to slaughter that animal and for that animal's meat to enter the conventional food chain, and it's in the grocery stores where I shop, and I buy that meat.
What color's the fat?
The fat's usually white.
Rodney, what do you think about all white fat versus brown fat in the meat?
So there's brown fat, there's white fat, and there's Sandy Lewis's favorite, yellow fat.
Yeah.
Okay, and yellow fat is created by carotenes, beta carotenes, and beta carotenes are in grasses and plants, and cattle that eat feed that's high in beta carotenes produce yellower fat. And some breeds genetically produce yellower fat. So all the Channel Island breeds of cattle have yellower fat as well. The brown fat that Rodney was talking about is not that fat. And he can elaborate on that more.
Right.
Did I answer your question?
Yes, but I do want to take issue with one thing that you said about the residues of antibiotics. And I am not sure that I'm the best person to speak to this, but with glyphosate being used to dry down so many crops, as we were discussing yesterday, and glyphosate residues being found in cereal grains and whatnot, even non-GMO crops, I wonder if anyone on the panel would want to speak to that. I mean, just a percentage.
And I wasn't talking about that particularly, and I think that's a valid question.
That is a very valid question. And that was my topic yesterday, and I guess it's going to be again today. See, that isn't, when we talk about antibiotics, everybody's leaving glyphosate out. And yet,
and fungicides as well.
And like Don Huber said, we're using, and like I said too, we're using 3 to 4 times more of that than all the other antibiotics put together. And then we don't even have it in our discussion. But when a mammal eats glyphosate, that includes us. You remember DDT, that Rachel Carson helped us get rid of, that went into the fat. So that built up in the food chain. So why I'm so disgusted as a veterinarian, most consumers get all worried about getting glyphosate through the meat they eat. But mammals, when we're eating cattle or pigs or rabbits or rats that they did their experiments on, 60% of the glyphosate that comes in goes right out the pooper. Which that's still a problem because it's a chelator, it cleaned up metal boilers, so now all my beef magazines are writing about the need to spend more money and use chelated trace minerals, because if you just use manganese oxide or zinc oxide instead of zinc methionine, it's taking some of them out. So we have to spend more money when we got it in there. Okay, so that leaves 40%, and then it's water soluble. So most of that gets peed out right away, or urine. It comes out in the urine. But in the trade secret information, and I think those juries got some of this, so whether cancer is down on our list, it isn't any more cancer causing than it's a risk. It's a risk. I shouldn't say cause. It's a risk for non-Hodgkin's lymphoma, but it isn't any worse than dicamba or 2,4-D or a lot of other things. So back to the 40% that gets urinated off, it's not all 40% gets urinated off. So when they look at some of the trade secret stuff, I think it was from 1983 to 1988, it's N-methylglycine phosphonate. So it's a man-made glycine derivative. Glycine's it's one of this amino acid, and it's a simple amino acid, so it lets proteins bend. And a lot of the enzymes in our bodies can't even fit into the cytoplasm of the cell if they don't bend. Same thing. But anyway, regrettably, industry knew, we approved it as a herbicide in '74, and industry knew that 0.5% of it, of this 0.5% of that 40% to 1.5% of that gets incorporated into our protein. And our protein making machinery, we got DNA and RNA that, our protein making machinery isn't all that fantastic. So when our bodies make proteins, a lot of times the first attempt at it, maybe 15 to 20, 25% of the amino acids aren't right. So we have these enzymes that use all these trace minerals that I alluded to that have to break that down so you can fix it. Well, it took them from 1983, I might not have the years right, but it took them 5 years outside of the body with synthetic chemicals to figure out how to extract that percent that gets bioaccumulated. So as a vet, I didn't want to listen when I was first working on this project and made these connections. And I've had a lot of help and some great mentors. Anyway, I didn't want to accept the fact that it bioaccumulated at all. So we're getting most of the glyphosate in our diet, and you said it, it's from this drying down, pre-harvest desiccation of crops right ahead of harvest. And that was never approved by anybody's government anywhere. It still isn't approved. Monsanto went worldwide and recommended it in 2002. And they're saying they're doing it for weed control. But weed control in the fall, when your oats, your barley, your wheat, your rye, your lentils, your peas, your peanuts, we use it on 70 different human edible crops. The Germans, it screwed up. The reason Monica Kruger got involved with it is because it's hard to brew beer when you spray your barley with it. And so the farmers over in Germany started spraying their barley with it in 2002. They didn't tell the breweries. So before Monica Kruger did any of her work in animals, she got hired by the breweries to figure out why they couldn't brew beer. So Germany outlawed spray drying of all non-Roundup crops, period, 9 years ago. The Netherlands outlawed it 2 years ago, and I'm trying to figure out why we're so stupid in the U.S. and Canada.
Well, part of that is a market. There's a market-driven reason behind that is further down the research chain. So when you use it as a desiccant, it increases shikimic acid, which is an ingredient in Tamiflu, which we have not had enough shikimic acid. And so I suspect, while I don't have any market analysis of that, if you go down the chain of how that's made and how that ingredient, so it's a high-value ingredient. So if you can increase the production of a high-value ingredient by doing something, the market drives those kinds of things.
Okay. Right. The thing that was not answered yet is this residue issue.
Okay, but we're going to...
Just a minute. This residue issue, the problem is that the labs are changing their detection levels, as Tyrone knows, as David particularly talked about. The way that you can say no detectable levels of antibiotics is that you drop or you raise the detection level so high that you guarantee you will not find that toxin. But as David talked about, it's the low levels combined that are really more toxic, not the high levels of a single product. So the industry can say no detectable levels of antibiotics, no detectable levels of glyphosate, no detectable levels of whatever pesticide they want to talk about, because they make sure that they raise the detection level by the laboratory high enough that they will cover most of the industry. So then they can claim no detectable levels.
So we're gonna move on to another one. For those who haven't seen it, the New York Times today has an article titled "Roundup Weed Killer is Blamed for Cancers, but Farmers Say It's Not Going Away." And all the farmers interviewed in it talk about how they need glyphosate and Roundup or they couldn't grow their weeds. It's an amazing article to see in the paper the same day we're having this conference. I'm going to let Mark do the next group.
Thank you all for this wonderfully elevated conversation about molecular biology and other things that I do not understand, but I've learned quite a lot about. I would like to shift our attention to the social sciences. This is an interdisciplinary conversation, and is anyone up here a social scientist?
All physicians are social scientists.
That's not true, but
Okay, most physicians.
still not true, but Alan, you're a social scientist. So my question to you, and I guess for everyone else, what role do various disciplines, anthropology, sociology, history, philosophy, how can they help you achieve the kinds of goals that we have been trying to imagine, or rather reimagine, in this conference? There's a lot of interdisciplinary talk, yet it's mostly scientists, industrialists, and physicians.
One way.
I wanted Alan to do this one first, though. Yes, yeah.
Okay.
You asked the best questions. Thank you.
Thank you.
So we've been over-focused on our intestines for the last 2 days. The biome is the entire Earth. We have this industrial agricultural biome between here and Colorado, which is an environmental disaster. We have the soil biome, which did not get enough attention, this complicated interaction between photosynthesis, air and water, and all the microbes and fungus underground that have this complementary relationship in terms of disease and water retention and delivering nutrients. And if we hold that thought and talk about Fremont, Nebraska, or Ottumwa, Iowa, or Bird City, Kansas, the idea of the rural economic biome, because human beings and families and communities are very much a part of this whole discussion, and we're not putting it in that context. There's a couple of things, because I don't want to belabor this, but there's a couple of things in a healthy rural economic biome, a community that allows and causes families to stay. Now, I'm going to throw out a bunch of things, and imagine if you didn't have any one of these in your life. A movie theater, elder care, K through 12, access to community college, a bank, hospitals, anybody else, a working water system, transportation, broadband, so you can actually stream a movie or do online learning. That's the top, that's the cream of 100 other things that serve the rural economic biome so that families want to stay there, so that there is economic vitality and jobs, opportunity, and hope. This big industrial economic system that we have, this concentration and consolidation, is like the antibiotic on that vitality. There are towns across the country, not just some, but the majority that are dying one family at a time, and people no longer can afford to live there, no longer want to live there, and they move around. The African American community has taken the brunt of this by far. The immigrant community, especially from Central America, that's been used to break up our small towns like Fremont and Ottumwa, they've taken the brunt of this by far. But we are, if you think about the gut, think about that massive, complex interaction of microbes and antagonists and protagonists and feedstock and the outcome. We are suffering in this country from a massive social dysbiosis. And I hope that in some way answers your question. But everything that we're talking about shows up at every level in this trophic system and all of these biome systems.
It actually sets the foundation actually really well for all of us to actually think about where we can utilize social researchers in biological research, right? How do we make sure, when we think about questions of equity, how do we make sure, for example, that the biobank is accessible to people in, Canarsie, Brooklyn? How do we make sure that certain kinds of individuals have access to these kinds of biotechnologies who might need them? Right? So as we think about these teams that we are creating, these interdisciplinary teams, right, can anyone think of anywhere in the research that you're doing right now, anyone, or in the field or whatever you're doing, can you think of a specific place where you think, oh, you know who might be good right now for this? If I had a community health researcher. Can you think of any moment or any place in what you do where these people might actually be useful for you?
Absolutely. And not only can I think of it, we have it in practice. We have an interdisciplinary practice where we have a PhD psychologist, a licensed clinical social worker, where we have, because my job is, going back to what we talked about yesterday.
And could you talk more, can you, describe very specifically what they do in terms of their roles and how it helps us think through this?
Yeah, I'm getting to that, which is to motivate people to make changes in their lives to improve their health. These people have a litany of obstacles to why they can and cannot make changes. We spoke of a couple of them yesterday as if they were one thing. We said people of lower economic resources and access as if they're one thing. They're not. There are plenty of people with that in our practice that have lower economic station, but they're spending a lot of money and their food stamps on the wrong things, and these other foods are absolutely accessible and covered. But they don't either know or want to make those changes. And this is where those socially educated people can come in, and we set up appointments or we work together as a unit to help the person have access, find ways of getting meals prepared, getting help in the home, whatever they need to undo these obstacles, many of which are from childhood traumas, are because they are handicapped in one way or another. So we have to make use of these people to help those folks overcome those obstacles. And pardon?
Holistic.
Yeah, it is the holistic approach in our holistic center. I have the Kansas City Holistic Center. So it is multidisciplinary.
I'm going to just make a quick comment about that. So I'm a physician, but I like to look at these sorts of things with a lot of humility. My cultural background is I'm from the Caribbean, and I currently live in the Southwest. There are a lot of Native American families there. And what I struggle with is how best to reach these cultural communities. If you're asking about research specifically, I think a wonderful area is what is the best way to reach people? I mean, some of the things that we found out in medicine is that, you know, giving people 3 different medications is virtually impossible for anyone to take. But combining a pill, combining different kinds of, for example, blood pressure medications into one pill might facilitate that. But how best do we reach the communities? That is a step back from just assuming that my cultural bias is the same as theirs in terms of, for example, a diet. It's not. Ask, you know, part of the experimentation, part of the research that needs to occur is how best do we communicate with each other?
If there's one message I think we can all take away from this, it's that we need to have education in learned skepticism. We made big mistakes. We accepted fast cures as answers rather than as a recognition that there was a symptom, an underlying thing that probably should have been fixed instead. Antibiotics as an answer is a mistake. Okay, it was a tool and it could have been used, and many of us are in this room because antibiotics saved our lives, but that doesn't mean they haven't been used improperly. And you know, Larry cautioned that the microbiome science is very early, but there's people in the room that think they want to go to the biobank from the Amazon and start taking cures now. Even your product that you spray on to reduce blood pressure, what if we find out 30 years from now that that causes Alzheimer's disease? Natural isn't always good, and we sure made a lot. So the economists need to help us solve this, the social scientists, and we need to teach skepticism, and we need to teach awareness at all levels, and the social scientists do a way better job of that than those of us in the sciences, really.
So there's a woman that I know who founded it's kind of a hybrid between a nonprofit and for-profit. It's called ZNA, and the Z stands for zip code, because your zip code is more indicative of your health state than your actual DNA. And she's working at the intersection of things that we heard about from David and Dr. Hayes today, and trying to work with public health and social systems, and this intersection of lots of public data sets that are available to start to be able to provide tools to the population, to the public health, to social scientists, to try to understand some of these complex environmental exposures, and how that is really what drives the zip code ZNA. So,
a couple things. One, great questions. I agree with that. The other one is, we think a lot about working with behavioral economists. A lot of our disease today is a result of the decisions that we make. And we think we are rational creatures, but that's not really true. We make decisions emotionally, and then we layer some reasons on top of it. You can go through a day without a meal, no problem. Try to go through a day without a rationalization. And the behavioral economists understand why we make decisions. And using that right now, marketers are very effective at getting us to buy stuff, the chronic use of which is we get sick. And we can think of lots of examples, mostly food, but other things too, cosmetics. One of my favorite examples is this is busily rewiring our brain while we're sitting here talking. 4 years after it was launched, automobile versus pedestrian in San Francisco spiked because nobody at peril of their life was watching where they were going. But it doesn't have to be that way. What we can do is try to engineer things so people make better decisions. And this is where, if we understand how we make these decisions, and the behavioral economists are really good at this, and besides, it looks like they have a lot of fun at work. If you're looking for some place to have fun at work, look at the work that Dan Arelli has done, and he's published a couple of books. It's very compelling. So we know how we can incentivize people to make better decisions. Right now, we use it to get you to buy shit that's very likely making you sick and you probably don't even need. Why don't we as a community who are interested in health use the same skills to get people to make better decisions? Look, we talked about medication there. Nobody, I don't practice medicine anymore, but I did, nobody takes their medication except 4 kinds of people. Drug addicts, they're very good at it. Obsessive compulsives, people married to obsessive compulsives, and those few people for whom taking their medication, missing their medication is potentially lethal, and only because they get selected out if they don't. Everyone else doesn't take them, and it's really hard to do this. So as a group, we know how to make people make bad decisions to buy products they don't need. Why don't we take that into, so it is a social science, but it's a social science of understanding how we make decisions and giving us incentives to make better and smarter decisions. What I'll tell you is I'm skeptical that people will make decisions because it's good for their health. They make decisions and they do things because it makes them feel good. Let's make them feel good by making healthy decisions, and let's figure out how to do that.
Can I follow up?
Yeah.
I could probably name off the top of my head 100 local small organizations that are doing the things that we're all talking about here. Growing food, developing community, healthcare access, multi-generational education, and making sure that the family jumps out of a cycle of economic and emotional depression and gets to a whole nother level. I'm going to break the rule against the extended metaphor, but in soil science, there's something called the Zonder mycorrhizae. And you can take dead dirt, and within dead dirt, there are thousands, if not millions, of isolated bacteria that are waiting to be connected. And once there's a plant, there's root, there's fungus, those Zonder mycorrhizae start to connect, and they start to form a quorum, they start to act in concert, and they are the fundamental building blocks of life in the soil once they're connected. And to really stress this metaphor, I'm all across the country. I can tell you people that are doing things that are locally relevant, locally built, that are deeply, deeply meaningful, all the way from central Denver to southwestern Georgia. And it's all about, to sum it up, 2 things: absolute inclusivity and access. The second thing is protecting that project, that motivation, that spirit from the larger system that wants to kill it off. And you can throw whatever metaphor you want at that. The last thing is that rural sociology used to be the cornerstone of every land-grant university. There's not a single rural sociology program left. And the old dudes who are the last of their generation of sociology, like John Eichert and John Comfort and people like that, are masterful writers. There's nothing new here. They've been there, they documented it, and they raised the alarm, and they were silenced. It's a whole parallel to this other issue with science being censored and controlled. The social sciences are equally vulnerable.
I just wanted to say something on the point of equity that you brought up briefly, and I'm not entirely certain how this relates to social sciences in the context of the question. But I just wanted to say generally, my takeaway from the last 2 days are we don't understand what's happening fully in the majority of the cells of our body. Is this correct? Right. This is a new frontier. And I think it's on all of us, and particularly those of us who are developing new technology and new ways of analyzing the microbiome and in medicine and whatnot, and in policy, how do we make sure that this new understanding, these new treatments that may be available to us in the coming years are available to everyone regardless of their educational status or their wealth or the race that they are. So I think we all just need to carry that, because don't need to repeat what we've seen in organics and other things like that, where it's something that's only reserved for a very white space and a very wealthy space. This is important to all of us, and we need to make sure that we're taking approaches that make this information widely available and the treatments affordable to those who need it.
Just a real short comment. We had the example of what was written in the paper where it was said that the farmers said they couldn't live without Roundup. And I looked through the group, and there was a defeatist look through everybody. And it's far from that. What we need to do is go and talk to those farmers, because I have talked to farmers that thought for sure they couldn't live without Roundup. The minute that I told them that Roundup was a very potent antibiotic, oh, we've been hoodwinked. We better have a look. There's another product called rotenone out there that they can use to cut down their crops. Wow, we're really thinking about that. I have a son-in-law that's an economist and is farming, and he's thinking about that because it costs him 4 times as much to use the other product. So education to these people. We can't be defeatist. Thank you.
And I just, I really want to say I appreciate all of the wonderful answers to this question, and to really think through something that, Belinda, that you had said, in doing this, and also in mentioning, for example, how new and novel this particular area is for all of us. It's vitally important that we make sure that on the journey of actually making these discoveries, that we are actually bringing all of these kinds of ideas and all of these people in other disciplines with us along for that ride. Because what tends to happen is, the projects are done, and then the social scientists come in and say, well, look at what we have here. And then they look back, and then the skepticism comes in. We need skepticism along the way.
I have a question for the gentleman on the end, all three of you. Oh, over there, Dr. Hayes and David and Alistair. From what you've heard over the last several days, or today, since you're not microbiome people, how is what you've heard today going to change how you think about the research you're doing or have done, and the way you talk to people about the implications of the research you're doing or have done, and in particular, the choice of things that you're looking for in the water, Alistair? And, I just would like you to address that.
Yeah, so I wouldn't say I'm not a microbiome guy. I mean, I talked about one bit of what we're doing, and one thing we are doing a lot on is soils and impacts of antibiotics and other chemicals on the soil microbiome and the functions of that microbiome. So we're doing a lot of work at the moment, for example, on wastewater irrigation. So the types of situation, which again I didn't really talk about, but I showed the photos of Israel. Now I guess here in the UK we're lucky because we have rain. Some might disagree, but in Israel they don't have the rainfall that we do in the UK. So they are very reliant on treated wastewater for growing their food. And I don't think there's any way around that. I mean, we are adding sort of irrigated water, which we know contains chemicals. The chemicals that they monitor at the moment, it's just a few obvious things. We know there's other things there, and we know those other things there probably are going to screw up the soil microbiology. And that will then have an impact on crop productivity, crop quality, and the stuff we're doing is showing that. So I think, as I say, it's something that we are sort of working on. I think sort of going forwards, the link to the human microbiome is something that probably I would like to get a little bit more in. And again, one of the things that I didn't show because I didn't have time. I could have spoke all afternoon if you'd given me the chance. But some other stuff we've done in Nigeria. We've most of what I showed was about urban rivers, and we actually went to rural locations where you have the village huts. The huts have pit latrines, and then within the village you have a well, and that well is providing the drinking water, the cooking water. Again, there's been a lot of work looking at the obvious contaminants in well water, but hardly anything on all these things that I'm interested in. And we find levels of antibiotics in the well water migrating from the latrines that are a lot higher than we see in our rivers in York. And those guys are drinking that stuff daily. And I think, that type of exposure, that's really interesting in terms of the gut microbiome. And that's sort of something that, sort of linking in with, people like Larry, could do some probably quite interesting stuff looking at that. What is that doing? Because I guess it's probably not a sensible thing to do. And one of those latrines was within 2 meters of the well, distance-wise, even though it was deep. So.
Actually, I am a microbiologist, microbiome researcher in the closet. My PhD was in microbial ecology. Most of my research, for example, right now I'm an expert witness in a case involving HIV transmission through a colonoscope. Most of the research I did was on how HIV, other pathogens survive inside the oral cavity and inside the gut. My current paper that I just submitted for publication is a lengthy commentary that makes the argument that the autism epidemic is real and it is driven by the Congress banning ocean dumping of sewage sludge, and then that triggering land applications of sewage sludge, and it brings all of my microbial ecology into the world of farming, what's going on, and again, how it impacts the gut. When I got into the oral cavity and HIV transmission in dentistry, it was all about how does this most fragile virus not only survive within the oral cavity, but actually hide away in that system. How does HPV survive when it's introduced by a flexible endoscope into the colon or the rectum? All of my work is about how different pathogens survive within the world that we're discussing here. So it's just, I guess, unfortunate that it surfaces in a way that it catches the attention of gastroenterologists and dentists, but it's actually all about what's going on in here. The most important thing I learned, and it was actually listed as part of a topic early on, how big should we think? The big picture that I've gained from here and before here is that I went about looking at local farms and how they were spreading sludge and how that was impacting the incidences of Staph aureus in the community and this sort of thing. It wasn't until I looked at some NASA photographs of their Earth observation system and saw that the little dust clouds I was studying from this municipality to that, blowing around here and there and impacting local communities. My gosh, you look at the Earth from a satellite and you see soil particles, dust from Western Africa streaming and visible, 1,000-mile wide streams of dust carrying all these infectious organisms, viruses and bacteria and fungi, moving across the Atlantic and settling in North America and South America and Central America. I could not understand when I looked at EPA's data on incidences of autism, which I was focusing on, which is all about the gut biome, understanding how I could take from the local farms here and there, coast to coast, Canada, et cetera, and translate that to the work that another component of EPA's Office of Research and Development was working on, which was the incidences of autism that took off in 1988. Well, 1988 is when Congress banned ocean dumping of sewage sludge. It went to the land. Looking at that from outer space, it suddenly made sense. When we talk about the biome and microbial ecology, we're looking at a planetary process. I could not understand how this part of EPA's data said worldwide, suddenly in late 1988, autism took off exponentially. How does that happen on a planetary scale? And how does it relate to what I'm looking at on this farm and that farm? All you have to do is look at that photograph. We are surrounded by an ocean of streams, continent to continent, of toxic chemicals raining down on us, infectious diseases. The whole controversial vaccine issue, nobody's even looking at the side of that is purely environmental. Every live virus put in that's attenuated and used in a vaccine, when it goes through the digestive tract, hits the gut microbiome, all those attenuated live viruses revert back to the original highly infectious rubella, for example. Rubella is defined as a vaccine-preventable infectious agent. It's vaccine-preventable, but when you get that vaccine and it goes through the gut biome, it's no longer attenuated. It goes out into the poop, it's spread all around us. So it drives me crazy when I'm watching the evening news and see an outbreak of measles in a theme park, and they know how it happened. Somebody didn't get vaccinated. When I took my kids to the theme parks and gritted my teeth and closed my eyes as they sat in little pools of water with 10,000 other children, most of which are shedding live measles virus because it's gone through their intestine, and they're sitting in this same pool with my kids and poops, creeping around.
Okay.
So it's, I'm sorry, but I'm a microbiome person too.
No, I mean, it's good. Okay. Tyrone.
Well, I'm not a microbiome person. However, I do, as I sit and listen to you and learn, I think about things like our mixtures issue with EPA was a study that we did combining everything that atrazine's mixed with on, there's no typical field. We chose one field that we were studying in particular. So I think there were 9 mixture of maybe 4 herbicides, 3 insecticides, and 2 fungicides. And we found that those low-level mixtures, among other things, resulted in an increased incidence of flavobacterial meningitis in the frogs. And our interpretation of it was that it was a negative effect on the immune system because all the animals, turns out, have the flavobacteria, the controls, the single, but now I think maybe it was a matter of the chemicals altering the microbiome that allowed the flavobacterium to flourish and actually induce a disease state. So it makes me think about some of the complexities as we try to move closer to doing real-world experiments, and these experiments where one chemical and everything else is controlled are nice and reveal a lot. But I think also we need to think more holistically, or I need to think more holistically about all the things that are going on in the environment when you alter and apply chemicals or antibiotics, as may be the case.
Well, thank you. So my last question, our last 10 minutes, is a very simple one. I'm interested to know, and we have students here who are taking a course called Big Ideas in Human Health, Disease, and Development. Yeah, shout out to my classroom. What's one big idea that you've walked away with, or that you will be walking away with from this conference for them, that you can share?
You asking the
No, I'm asking you guys.
I'd love to know what they.
Yeah, I
Well,
think that's a
or
great.
you know what? Ooh, man, wow, remember, I didn't do it to you. They did. Earning those participation points.
Well, I forgot who, but somebody mentioned antibiotics and kind of the overall effect of it. I'm interested in antibiotics as a whole because there are many side effects with antibiotics, such as giving certain diseases, such as MRSA, the ability to be exposed to these stresses to pathogenic bacteria, and giving them the ability to adapt to various antibiotics, and along with the pollution of the oceans. So I was just wondering, how can we safely eliminate pathogenic bacteria, or what actions are being taken towards that goal? Because to me it seems as if antibiotics, it's posed as an overall solution, but it's actually kind of a problem in a way.
Well, antibiotics are, I think, you weren't here, I think, yesterday, or maybe you were, the discussion around antibiotics most are produced by bacteria. And they are, one of the communication tools that bacteria use. And when we talk about pathogenic bacteria, I think that's also that we will learn that we're thinking about that a little bit out of context as well, because it's the environment that drives the behavior of the organism. And when you have a healthy ecosystem, there will be all kinds of organisms, many of which may be in that system or passing through that system, that would have been considered under something called Koch's postulate of, one pathogen, one disease, that will just pass through and not cause a problem. So it is a lot about the environment, from an antibiotic perspective, and from a microorganism perspective, and from a societal perspective. What is the environment that we're setting up, back to your social question, that is creating a pathogenic behavior in our society, a lack of respect for each other, and the loss of our ability to communicate and work together. Because back to Alan's point,
The same thing is happening in our microbiome. They're losing the ability to work together. We're losing that ability to work together. So I hope that the conference has inspired people to take away some thought about that, about how can we rebuild this complex network and work together.
The other thing about this is that getting rid of antibiotics means building a strong immune system, yes, through the microbiota, absolutely. But there are so much of our biochemistry other than and because of the microbiota that we're deficient in today. A strong immune system is built with adequate levels of vitamin D, adequate levels of vitamin C, adequate levels of beta carotene, of vitamin A, vitamin E, as we found out from the Olympiad athletes. We can't just keep killing. One other point I would like to bring up that I kept thinking would be brought up here at this conference is a real clinical hardship, something that is very common now. It is also misdiagnosed. It's been used as a trash dump by GI docs that have heard of it and don't know what to do about it, and that is the advent of small intestine bacterial overgrowth, whereby we have the growth of our good microbiota in the small intestine and the tummy. So the moment you eat anything that's healthy, that has butyric acid, that is fibrous in nature, or that has soluble fibers in it, your tummy bloats up within 5 minutes of that meal with extreme pain, and these people end up migrating to a meat and potatoes diet because that kills the microbiota, and so they can be comfortable. This is one of the problems we've had with antibiotics. We know that this happens too because when we give an antibiotic, particularly a Z-Pak, if everybody heard of a Z-Pak out there? You have a Z-Pak, it will take out that upper level macrobiota. This part of our intestinal tract is relatively sterile compared to the colon. The role of the appendix is to re-inoculate the gut when the biotic count goes down. Your appendix it's not junk mail. It has an important purpose in our bodies.
I just want to, I am very fascinated to still hear this, but I do want to make sure that we give time to the students to answer this particular question, to ask questions to our panelists about, their big ideas.
Okay.
Henry.
Okay. Hi, everyone. My name's Ismail. And
sorry,
just want to be clear.
Ismail. Henry, you're next. I'm next. Well, yes, because you raised your hand. So Ismail, then Henry. I was looking there and said Henry's name.
Oh, no, sure, okay. So we learned an interesting word the other day, biopolitics.
Oh, dear.
I'm sure you're all familiar with. I know, right? I wonder who taught me that word. And something that I think that we're all thinking about, and I keep hearing this from the panel, which is something. It's always when you're talking about the future and you want us to fix something. You give us problems. You do. You want us to fix things. And I think what we haven't talked about is how specific political structures are constantly in play with how these projects are being run, and how these works globally are being affected, and how communications can be interrupted between scientists who need to be in communication with each other to get these projects done, and how certain people with lots of pull decide that global warming doesn't exist. And we need to decide as children Just to clarify, because just because we're 17, doesn't mean we're just gonna go pop off with like adult responsibilities, because we really are children. Haven't been to college yet, even though we're in one. And we need to fix something like global warming, which, generations before you and your generation have together create a massive problem for us. And I think that we should be talking more about how political structures are affecting your work and are problems for your work, instead of just saying, well, like there are different social, scientific issues that like we haven't addressed and we don't have the answers. We have all these questions of microbiology. And, we just want to know if you can hand us a baton that isn't like completely deteriorated, and you can hand us like pieces of things that we can like put together, and then like teach us how to do that, just so we have a chance to like, move forward, instead of like several years of regression. I mean, I'm just like, I'm curious. Yeah, no, yeah, you get it. It's wonderful. Yes, love that. We love that.
Well, I think one of the things before you take the baton, make sure you get your work boots off and put your running shoes on. And that's learning. You have to learn what's in the political system. And I think probably a lot of what's taught is useless. No, I'm serious. I was a grad student at Davis, and I was lucky enough to get an internship with the Legislative Analyst's Office, and they were an exempt group that looked at the budgetary and the policy issues that were being presented to the legislature and to the general government. And I had no clue about all the backroom games that we're playing. I watched senators in policy groups and fiscal groups laughing and spinning around on their big chairs because they'd screwed the people, and they were so proud of it. So, I mean, there's stuff like this that is going on constantly, but you'd never see it in the newspaper, you never see it in the magazines. So one of the things I would suggest you invest in is a good internship, so you begin to understand that. And also look up and get a good grounding on politics as a mechanism of clientele capture. What agencies are prone to clientele capture, whether or not they're given critical things to do that they'll never be able to do because it looks good. And so those would be the two things that I would suggest you do: an internship, good running shoes, and well, I gave, maybe you want to know about that. I was the environmental officer for 22 countries in Africa with USAID. And at the end of that, you had an end of tour report to hand in. So mine's about 2 inches thick. It's on a thumb drive, and Roger has that.
He just gave you homework.
Thank you so much.
Science is political. I wish someone had told me that early on. I didn't learn it until late.
David?
Science is political. And so just keep that in mind, that, we can have our idealism about this, but if we want to have impact, we just have to accept that as part of the process.
I mentioned earlier the commentary, two-page commentary I published in Nature, 1996, titled EPA Science Casualty of Election Politics. I spent probably 6-7 years filing whistleblower complaints against the EPA, not to make money or anything, but it gave me discovery power. I wanted to know, I wanted to understand more than science. The most important thing to me was to understand the nuts and bolts of how do we pass Clean Air, Clean Water Acts, but no Clean Soil Act, no comparable protection for the soil, and then the powers that be who have cleaned up the air and the water then want to concentrate everything and apply it around us, where we live and work and play, where we raise our children. How does that happen politically? Because it's a politically created problem. So I went after, from the top up, as far as I could, which was to Carol Browner, the EPA administrator, who 100% supported me. She told me that at an award ceremony after she gave me the Science Achievement Award for the same paper I got fired for. And she was overseeing Jerry Melillo, who was working with me on climate change out of the White House. In fact, I funded Jerry Melillo to do his research, and I did it with him on climate change-related gases. And I asked Jerry Melillo one day at breakfast, I said, Jerry, you ever talk to Carol Browner about what's being done to me over this really big problem I'm trying to solve? And he said, David, I have been around and around and around with her, and all she will say is that she's overruled at the highest level. That was what I was looking for 4 years. How high does the politics of this go? The EPA administrators telling my contact with the White House that it's the President of the United States who is pushing the buttons on this stuff. It is that high level of politics. The only way these kind of problems can be fixed, and I'm so happy you asked this question, because if you don't understand the politics, it can't be fixed. The problems we're discussing here, take my part, sludge, which I look at as equivalent in every way to climate change, but nobody's doing anything about it. The only way to fix that problem is to have somebody like me who understands not only the science that's involved from top to bottom on every scale, from a local farm to planet Earth, understands the science, but also understands the politics within federal agencies. Dwight Eisenhower said on his way out in 1961 that thanks to federal funding of academia in the United States, the university system is no longer a fountainhead of free ideas and good science. He says money is what's driving it. So if you understand the science, or at least have 2 or 3 people together who really understand the science at every scale of one of these problems, but also understands federal bureaucracies, how they work. I know how policies get developed and enforced within EPA, the USDA, the FDA, the CDC. I can take these problems if I was at the top, which I'll never be the President of the United States, but if I had the ear of the President. When the polio epidemic was being dealt with, I think it was FDR that sided up with one of the scientists developing vaccines and got behind that scientist. That scientist was like me, only on polio, though I do look at polio a lot. But the president got behind that scientist, and whatever that scientist wanted, the president made it happen. Nobody could get in the way. That's the only way to solve this problem. I know the scientific solution to sewage sludge. There is only one solution. All these sewage treatment plants that are producing the sewage sludge that now contains all the infectious microorganisms that we're trying to deal with, that we have vaccines for, and every toxic chemical there is in the universe, it's all there. It's a simple matter of taking that material, raising it to a high enough temperature, which is done under gasification, where you break down all these toxic chemicals to carbonates, phosphates, you know, the building blocks of life. And you take all that carbon that's in there and you activate it. So it binds the traces of heavy metals, of lead, cadmium, arsenic, mercury. And then you can put it on the land like the indigenous people did, the populations that came across at the end of the last ice age and made it down to South America, to the Amazonian region, which was not fertile then. But they took bone and cattle dung and cooked it and began to till it into that soil such that they created the Amazonian forest, which is the richest, most diverse area on Earth. Provides 20% of our oxygen we breathe. And that system, which they created over a few thousand years, sustains itself to this day. So we've got to connect the politicians, and it takes a rogue politician at the top. We need a rogue president with brains.
All right.
If we could ever achieve that.
And on that note, I would like to bring us to our closing. And
yeah, I've said too much.
That's a great way to end. Hello, everyone. My name is Malia Dumont. I'm Chief of Staff here at BARD. I'm Vice President for Strategy and Policy. I wasn't able to join you yesterday, but I've been listening in all today. And very glad to have this very distinguished, unique group of people coming together, and especially to have some of our students here in the room. It's a really wonderful multi-generation event. And on behalf of President Botstein, I want to thank all of you for participating in this. Roger, unfortunately, he had to leave a few minutes ago, so he asked me to step in and say a few words. First of all, to thank you all. Second of all, to mention that if you've enjoyed this event, this is just one of many that the Hannah Arendt Center puts on a regular basis. Our annual conference is next month. That's what these flyers that you have on your seats for about this year's conference is about racism and anti-Semitism. So we don't shy away from the difficult issues. And we look forward to really nuanced, complex discussions. On the topic of this particular gathering, we've been recording it. Video will be available, and we'll follow up to provide you the link for that. And we think this has been the beginning of a really rich discussion that's worth taking forward. So we're going to look into how we can continue this discussion, maybe have a follow-on meeting in the future with a similar group of people, and we hope all of you will stay engaged in that work with us. So once again, thank you very much, and safe travels to all of you.